recording. Okay, we're ready. So today we're having our next guest. Dr. Andreeva is going to talk about her research, about her lab, about molecular mechanisms of diseases. But first, I'm going to give you the floor, and at the end, we're going to have a little Q&A session where students can ask some questions about what is interesting, maybe some tips about research, some insights. Okay, so floor is yours. I'm going to give you a co-host so that you can share your screen. Okay, so I believe you should be able to do that now.
Okay, yeah. Can you see my screen?
Yes, it's in.
Can you see? Okay, good. So we can start. So before we start, I would like to outline what I would like to talk to, so I wouldn't ruin your expectation, what my actual presentation would focus on. So initially, I will talk a little bit about my lab and specifically about 3 people who work with me. Then I will talk a little bit about the criteria I use when I consider to take the student to my lab or not. So the reason I'm going to talk about it is that a lot of students send me emails and ask to kind of take into the lab. And I sometimes answer those emails. I really have a lot of them. And I think it would be easier if I tell criteria first so everybody would know actually who I'm looking for. And then after that, I will talk a little bit about the project. I will give you a general idea about the project. I will not give you really a lot of specifics because those are ongoing projects. They're not published yet. And I'm really uncomfortable having something on the YouTube, which I have not kind of reevaluated and have not published yet. So, but I will give you reference to 2 of the recent papers where I'm a co-author. So if you are really interested, you can read the papers and get specifics from there. And I will also kind of outline general direction I'm going to in my lab and what methods I'm trying to establish. So first of all, the lab name is Laboratory of Molecular Mechanisms of Diseases. And this name of the lab is really a result of some bureaucratic process. So I and Darhan, we were jumped in a really small room together. So we are sharing the lab, and we were requested by the administration to give this lab a name which would kind of encompass all topics that we are studying. That's where the name comes from. It's very unspecific. It's hard to conclude what we are going here and what we are doing. So, but you can get an idea that what we study biological processes on molecular level. We are mostly focused on the level of the proteins, on kind of level of individual proteins. And all proteins that we study are implicated in certain human diseases or disorders. So the name reflects this fact. And so lab is located in C4 building, room 643. I believe Darhan showed you this image already. And so I don't have really entire lab. All I have in this lab is this small bench. That's my bench, and that's it. So I'm using this bench, I'm using this hood to grow cells. And so that should tell you immediately that we have quite limited capacity. And I'm very careful about who I get into the lab because we're already pretty much crowded. So far, I have several people who work with me. And so one of those people is Aralim. And she's Ray. She just joined the labs a couple of weeks ago, and she's learning all methods. And if I take a student in the future, she would be probably the one who would stay with the student and help the student to learn new kind of simple methods and procedure. So I also have Arai. I'm really fortunate of having Arai. She's a 3rd-year biology student. Very hardworking. She comes to the lab each day and she does a lot of different small projects. The project mostly she's focused right now, we are trying to establish purification of procedures for purification of certain proteins which are implicated in the hearing and are important for our ability to hear. And so what she's trying to establish is the procedure for purification of those proteins, for preparation of samples for mass spectrometry analysis, so studies covalent modifications and identify maybe new covalent modifications for future validation in the in vivo models. And also Tamiris, who is actually a fourth year chemistry student, she's currently also helping me a little bit with biochemistry projects. She's mostly involved in the purification of specific proteins and some cloning projects. So I also work with some collaborators from NCB, National Center for Biotechnology, and like Pavel, and he helps me to do mass spectrometry analysis of my samples that we make together with RI. So those are three people who work with me right now, and I would like to spend some time clarifying, because despite the fact that we have a very small space, I'm quite open to the new students to join the lab. However, I would like to warn everybody that type of research I'm doing is I'm trying to do really high quality of research. It means I would not publish anything that is sloppy or crappy. We are trying to establish methods and procedure that would give us kind of perfect result, not maybe entirely perfect, but on the level that would be exceptional to the high impact journals. And so for us, it takes at least 3 years to do publication and a lot of hard work. So if a student joins the lab, from the day that the student joins the lab, it will take probably at least 2-3 months for the student just to get hands-on basics procedure, and it will take maybe 3 years before we would be able to publish the results of this student who joined the lab. That means immediately that if a student who joins the lab wants to have the publication before the graduation day, that means students should join at the end of the first year. So, and that's normally what I tell everybody, like if you're a first year student and you finish your exams at the end of the April, then first day May, you can join the lab, and May, June, July, I will train you with all procedures. We will work each day, and I should make sure that you can not just follow protocol, but follow it mindfully and do all experiments at kind of high standards. And once you learn all these methods and approach, then even when the year starts and I wouldn't be able to supervise you each day in the lab, if you learned all methods, you would be able to continue to work on your own during the next year, maybe 2 years. And so that means that the student who joins the lab should plan kind of working in the lab for 2 years. And the good start is the end of the first year, the beginning of May. So, and that's true for students who actually want to leave university. There are a lot of students who actually plan to do PhD programs abroad. And so we plan from the start that after the year, they go abroad. And so for those students, I say first year is when you want to start. If you're a third year student and you don't plan to go abroad and you plan actually apply our master's program, that means you also have 3 years, your fourth year and 2 years of the master program. And so you can also join me in the May at the end of the third year and work for the fourth year and masters. And then that means at the end of your master's project, you will have a publication with you probably as a first author. But students should consider very carefully, because that would be 3 years commitment of day working in the lab. And I know not everybody is able to fulfill this commitment, and it might be not possible for everybody. However, this are some of my criteria, and I would like to tell kind of upfront to students that I expect them to work for 3 years before publication. And so now to switch to research areas, we have broad interests, rather broad, and who took my courses, some of them might know that I'm interested in the cell signaling and specifically at the cell cycle control, and not from the perspective of the cancer when cells go through the cell division uncontrollably, but actually from the opposite perspective is when cells are actually unable to re-enter cell cycle. We are normally talking about highly differentiated cells in this context. They're not able to re-enter cell cycle and they're not able to undergo cell divisions. And so I look at them from the perspective of the regeneration of the tissues. So we all know that certain tissues, human organism, once they're damaged, humans are not able to regenerate those tissues. And the list is quite extensive. And so some of those tissues are actually sensory epithelia that are responsible for our ability to hear or for our sense of balance. And so I'm mostly focused on those types of the tissues and how we can actually circumvent the problem the fact that we humans are not capable of regenerating the tissues. So that's our kind of general perspective, and I would like to say we use a lot of protein chemistry. So we work on the level of in vivo and we work on the level of in vitro. So we do protein chemistry, and regarding these types of techniques, I would like to mention first technique I'm not gonna cover, like first project, I'm not gonna mention it at all because I'm doing it in the collaboration with my colleagues in American universities, so I'm not about, but I would like only to say that what we do here, we purify proteins and we use mass spectrometry analysis to identify potential covalent modifications, and we study how these potential or kind of real covalent modifications affect protein-protein interactions. Well, yes, so that's one of the perspectives. And so another type of techniques where we use a lot of protein chemistry and actually, perspective or kind of what we do in the lab was already covered by Darhan. I believe a couple of weeks ago, he gave a presentation on the lab tour where he actually described in details the project which was published in this paper. So we did this project together. I don't think it makes sense for me to repeat what he said. I only would like to mention specifically what techniques I actually do in the lab, and we already established and students can learn when they join the lab, which might be kind of helpful for their future career. So we do a lot of cloning in the labs. We are quite comfortable designing and cloning a lot of different constructs. And one of the methods that we use also CRISPR gene editing to create homozygous gene knockouts. And so I just want to use a figure from this paper to illustrate the point that this method is established very well in the lab, and we can do it quite successfully. And so what you see in this slide is a Western blot analysis of different cell clones that we created by using this CRISPR gene editing technique. And so 2 control clones and 2 homozygous gene knockouts, where we were able to knock out PARC7 or DG1 protein, actually gene. So, and we use CRISPR technique. Those of you who are familiar with CRISPR technique, you know, special endonuclease involved that would actually introduce nicks in the double-stranded DNA, and that would result in the creation of indels. Those nicks are normally introduced at the -3 position from the PAM sequence, which is shown here, TGG. And so if you're using this technique, you know where to expect indels, and you know where to expect mutations to happen. So we use this technique, and we use gene sequencing analysis. So here you can see result of sequencing of the same exome sequence from the control and from the knockout. And I hope you can appreciate that in the control, the sequencing is quite very well defined. But in knockout, strictly at the position of -3 from the PAM sequence, that's the position where Cas9 endonuclease is supposed to create those indels. Actually, nicks that lead to indels, you can see you have two different sequences and they don't match the original sequence. That means that both chromosomes get these nicks and indels were created in both chromosomes. And so sequence was altered very often. That leads to the mRNA degradation. And as you can see from the Western blot analysis, and that's another technique that is very well established in the lab, we were able to kind of get complete knockout for the DG1 protein. On the bottom is GAPDH control that shows you we loaded equal amount of the protein when we analyze this sample. So to sum it up, we have quite well-established gene cloning techniques, Western blot analysis, sequencing, cell transfection techniques, which are very basic techniques for anybody who would like to do research in biology, and you can learn them in the lab. There are also some techniques which are not established yet, and I'm just trying to establish them. So I'm in the processes of establishing several techniques which are actually in vivo technique or explant cultures. And those are far more complicated than these basic techniques and require a lot of initial investment. So if you as a student would want to learn these techniques, you can count on that it will take you at least half of the year just to be able to perform these procedures. So they are not simple, so they take a lot of time. Some of them, those experiments and procedures that I would show you would require you to stay all day long, starting from early morning till maybe 12 in the night, to be able to complete required steps at the beginning of experiments. It's just one day, and generally experiment lasts several weeks. So once you started this experiment, you cannot stop until you finished. So that means you need to work 2 weeks at least, days, several hours. And so those techniques are not well established. So, but I would like you to get an idea what I'm actually trying to do and what this project is dedicated to. And to illustrate it, I'm going to show you images from the techniques, same techniques that I did at the university where I worked before, at the University of Virginia. And just to give you an idea what I'm trying to achieve here. And I would like to mention that pictures, most of the pictures that I'm going to show you and most experiments were done at the University of Virginia. And I had a wonderful opportunity to do them in the lab of Jeff Corwin. And I initially did that project, but then I moved to Nazarbayev University, and Mark Rudolph was actually a graduate student who was able to finish this project and bring it to full completion and publish the paper. I would like to reference everybody to this paper. So if you want to go through details, everything I'm not going to cover everything here, but I would like just to give you a general idea what this paper is about. Again, so if you are interested, just go and read the paper. It's very well written and it's easy to follow. So this paper, as you can see, about hair cell regeneration. So anybody knows what is hair cells? What kind of hair cells we are talking here about? So in this context, these hair cells have nothing to do with the cells that required to grow our hair. In this case, these are quite specific cells, epithelial cells, cells of epithelial origin that can be found in sensory tissues in the ear. They have quite a distinctive morphology. They have these stereociliary bundles, quite tall actin filled protrusions, which have quite characteristic morphology. And because of the presence of those bundles, they had the hair cells. And so those are the cells that are able to convert mechanical stimuli into the neuronal impulses that can be interpreted by our brain. And those cells are absolutely essential for our ability to hear or our sense of balance. So without these hair cells that actually can convert one type of energy into another one, you would not be able to hear or have some balance. And so here you can see isolated hair cell in this image. And so, but it's kind of wrong impression. Those hair cells can be found in the patches of the sensory epithelium. Here you can see a schematic representation of the organ of Corti, that the epithelial tissue that is responsible for our sense of hearing. And those hair cells here are color-coded in violet color, and I hope you observe the fact that they don't exist in isolation. They are surrounded by another epithelial cells, which are called supporting cells. And here you can see kind of side view of this organ of Corti, and you can see top view, like bird eye view, and I hope you appreciate that each hair cells has this crown of stereocilia, and that's where the name comes from. And what I've done, I'm quite comfortable of isolating those specialized sensory epithelial tissues. So I know how to do it. And so that's one of the techniques that I'm trying to establish here. So here can another example of sensory epithelia that actually responsible for our sense of balance. In this case, that's utricular utricle, a schematic representation of utricle. And I hope you appreciate the fact that it also has hair cells interdigitated by supporting cells, color-coded here in orange. So what are those hair cells are responsible for, and why do they actually has this stereociliary bundles? So those stereociliary bundles is the organelle that actually required for this mechanotransduction. I'm not gonna go to the details, and I'm not gonna explain everything here on molecular level. I would like just to say that during this process of mechanotransduction, what happens, those stereocilia get deflected, and they can be deflected either toward the taller straw of those stereocilia or toward the short straw. And depending on the kind of direction of deflection, the mechanotransduction channels would be either open or closed. And so when they're open, that would allow depolarization, that would allow the flow of ions, that would lead to the depolarization of the plasma membrane, and that would eventually lead to the release of neurotransmitter and stimulation of the neurons that would send signal to our brain. So those hair cells are absolutely required for this mechanotransduction process. And so we humans, for example, are born with two inner ears, with two sensory epithelia, organs of Corti, and each organ of Corti would have only 14,000 of those hair cells. And what is remarkable about them is that once you're born with those hair cells, they will stay with you for life. And so we humans do not have any ability to produce new hair cells during our lifetime. And so if those hair cells are damaged and they die, and they can be really easily damaged by loud noise, by certain antibiotics, aminoglycoside antibiotics that are used to treat certain conditions, by certain drugs like cisplatin that are used to treat cancer. So once they're damaged and they die, they cannot be regenerated by us. And that would lead to the complete loss of hearing. So you would not be able to restore this process. So that's actually quite tragic. So we normally refer to this type of hearing loss, sensorineural hearing loss. And here you can again see this list of causes like loud noise, antibiotics, some cancer drugs, and simply aging. Just old people have quite a lot of those cells damaged and either gone or dysfunctional. And so once this happens, there is no way back. And it's estimated that hearing loss, so at least 5% of the world population, and you can think about it as 466 million people actually suffer from disabling hearing loss. And it's not just very expensive, it costs us a lot of money, but it's actually quite tragic because this type of disability, deafness, people think that it's far worse than, for example, losing your ability to see your sight. Because once you lose your hearing, you lose all connection with people. You are not able to communicate with people, and you become quite isolated. So there are far higher percent of depressed people who commit suicide among deaf people than among people who lost their sight. So why I'm bringing this up is that we humans cannot regenerate those hair cells once they are gone. But what's remarkable, our organisms are quite capable of doing this. What I'm showing it to you is actually sensory epithelia from the chicken, from bird. And so here you can see a lot of hair cells in chicken cochlea, that's organ responsible for the hearing. And here you can see this cochlea after the damage of the hair cells. So chickens were exposed to really loud noise. They lost their hair cells, and you can do a test and prove that those chickens are not able to hear very well. But after like 10, 12 days, they are able to regenerate their hair cells and restore their hearing ability. So a lot of animals are capable of doing it. Birds, reptiles, amphibians, fish, shark, so have this regeneration capacity, but mammals don't. And so that brings us to the point and to the question, what actually happened? Why we lose this regeneration capacity? And so you can answer this question if you compare your control, for example, birds who are able to regenerate. What happens during this process of regeneration? Who is responsible for regeneration? And studied there. And that was done actually quite a while ago. Here you can see just image from the review paper. So what we know from those studies, it's actually supporting cells that surround hair cells are responsible for this regeneration. And so when the hair cell, for example, is damaged by treatment with antibiotics like neomycin, it will die and will be extruded. And so that will lead, that will trigger cell cycle reentry. From inside of the supporting cells, they can re-enter cell cycle and divide. And so after the division, one of the cell will remain supporting cell, and one cell would be able to differentiate into the hair cell, and that would restore your hearing capacity. On the other hand, sometimes supporting cells can directly convert and differentiate into the hair cell without the division. So both routes are possible, and we know that a lot of organisms are capable of triggering this cell-cell cycle reentry in the supporting cells, but we humans don't. And so here brings us to the point and to the question, why we actually lost this ability during evolution, why all other animals on this evolution tree are able to do this, and we humans or mammals, we can't. And what happened along the way, so what went wrong? And we know now that a lot of things actually changed, so there is no simple answer to this question. There are multiple events, and probably it's a consequence of different inputs. But what we know is that we actually can establish certain models and try to kind of find out what are molecular mechanisms that prevent this regeneration in mammals. And so one of those models that I used in the Corvin's labs and I tried to establish here at the Nazarbayev University, the model where you can use utricle epithelium, and you can either, for example, trigger cell death in those utricles and study whether those utricle supporting cells are able to reenter cell cycle and trigger regeneration of the tissue. Or you can do what I've done here. I did explants of the utricles and assessed different stages of the utricles, whether they're able to reenter cell cycle or not. So what you see here, two utricle tissues, two utricle epithelia at different developmental stages. So what we know, and I would like to mention, so why utricles are unique. So utricles at early developmental stages, P0, that means it's a utricle from just newly born mice. It was collected and dissected from the mice that was just born. And so those utricles and supporting cells in these mammalian tissues still retain ability to regenerate and to reenter cell cycle. That's what this experiment actually illustrates. So once you isolate this tissue and plate it on the stiff surface, that will trigger the expansion of the tissue will start to spread and those supporting cells will reenter cell cycle. You can estimate it by the incorporation of BRDU into the DNA of the cells, which would reenter S phase and will start the replication. And you can see here that's the same utricle tissue, but just mice, which is 9-old. And as you can see, there's not much of the spreading of the tissue, and supporting cells are not capable anymore to reenter cell cycle and trigger cell division. And so what this slide is supposed to illustrate that those utricles gives us opportunity to study two different conditions. One condition where the tissue still retains capacity to regenerate, condition when it completely lost this capacity. And so we can compare these two conditions, use it as a model to find out what actually changed. And so by using this type of model, that's what I'm trying to do. I'm trying to uncover molecular mechanisms that restrict this proliferation capacity in supporting cells of the mammals. And I don't know how much time we have left. How much time we have left?
25 minutes at least.
25, okay. So I'm gonna go quickly and just show you what techniques I'm trying to establish, and I'm quite confident I would be able to establish them in my labs. So I would be able to teach students. So if would like, for example, some would like to do master's project with me, or graduation project and then master's project. So I'm, for example, quite capable of isolating both cochlear and utical tissues. It's an intensive labor process, but you can learn it. It will take probably a couple of months, but you can learn it. And so what you can see here is the cochlear tissue. That's the one that's responsible for our hearing. And so we can dissect it and do monofluorescent staining and do specific analysis for certain proteins for their localization and maybe other specific outcomes that we can use as a report. So here you can see it's another type of procedure that I'm trying to establish. This one is dissection of the utricles. Utical tissues from the mice at different stages. So this one is, for example, utical from the newly born mice. That's one that still regains capacity to regenerate, and even earlier stages. Those are the stages where actually supporting cells naturally still undergo cell division, and you don't need to kind of even to trigger this process. You can just use those tissues to study it, natural kind of settings and environment. And we can easily also isolate utricular tissues of the older mice. P16 is the stage, that means P16, it's the day 16 after birth, when those utricles completely lose their ability to regenerate. So I normally try to compare P0 utricles to later stages if I'm doing explant and delamination types, or I can, for example, compare P16 with E16 or P0 utricles. So this gives opportunity to have two conditions. One condition where we have regeneration of the tissue, and one that's capable to regenerate, and compare them. So I'm doing the same, what I just showed you, some explant cultures, and I'm trying kind of use this technique to assess a lot of parameters. And so the way how we determine whether cells were able to re-enter cell cycle and divide is by using this BRDU assay analysis. And this one is rather simple, so everybody also can learn it. So here I can show you images. This image was unlike the previous one, actually taken here at the Nazarbayev University. And one of the students in the lab, Gaziza Yiltai, she's now in medical school, did this BRDU incorporation analysis, which is quite standard technique, and you can learn it in the lab too. And so what I would like to say from this perspective is that this project was initially initiated in the University of Virginia. And so the paper was published, to which I kind of reference you, but I'm still continue this project, but by using probably or studying different molecular mechanisms. But I would like to use specific molecular mechanism that is related to the HIPAA pathway and one of its downstream targets, YAPTAZ, which is transcriptional co-activator for the T transcriptional factors. Just to illustrate you kind of the logic that we use here and try to manipulate. So here, that you can read in more details in the papers. I'm gonna just use it as example. That's what we did, and I'm comfortable showing it because it was published. So the logic was like this. So we hypothesized this mechanism might be involved and altered in mammals, and because this molecular pathway, which involves YAPTAZ TD-mediated transcription, is altered. That's why utricles or other sensory epithelia are not able to regenerate hair cells. And so once we hypothesize it, we actually test it in the paper whether it's true. And we actually proved that that's probably one of the mechanisms. It's not the only, but just one of the mechanisms. And if you prove it, if you test and it turns out indeed that might be the cause, then you can actually try to manipulate this pathway artificially by using some tricks, molecular tricks, and try to manipulate it and actually force those supporting cells to re-enter this cell cycle artificially. And that's what was done. So why actually picked up YAPTAZ TAD? There was a lot of logic behind it. I don't have time to explain it, but I would like to mention just one thing. So YAP, which is transcriptional co-activator, it can bind TAD. YAP is not able to bind DNA directly, so it binds to the transcriptional factor TAD. Normally together they bind at enhancer regions and regulate the transcription of genes, which is mediated by polymerase 2. And so what you see on this slide highlighted in red are just certain genes which transcription can be regulated by YAPTAZ TAD complex. And if you know a little bit biology, you quickly realize those are the genes that relate different stages of cell cycle. So in fact, really huge percent of the genes that regulate cell cycle, the expression can be regulated by YAPTAZ and TAD. So that was one of the reasons why I focused on this specific pathway, but not the only. So there were also a lot of other reasons. And so the logic was, if it's really true, if it's because this pathway is altered, can we artificially kind of stimulate this YAP-TAD dependent transcription to trigger cell cycle reentry. And so indeed, we were able to do this. And this experiment and this figure just illustrates this point. So what you see here, I would like to go a little bit into the details to explain you so you won't be confused. So here you can see 4 different conditions. You can see 4 different utricles. So, and these utricles were infected with different viruses. Specifically, those are adenoviruses, and those are adenoviruses which were altered. So we just use them as vehicles to kind of artificially inject cells with genes that we would like to express. In this case, we were expressing different YAP constructs. And so what's remarkable about this adenovirus system, it specifically infects only supporting cells. Adenoviruses infect hair cells at much lower efficiency. And supporting cells are incredibly difficult to transfect. So, and this technique is one of the few that gives you opportunity actually to alter artificially expression profile for certain genes. Now, I'd like to mention that at Nazarbayev University, we are not allowed, so we don't have any facility with the safe level that would allow us to work with adenoviruses. So this technique is not available for us. And so that was done specifically at the University of Virginia, but I would like to mention that very soon we will have new microscope, and we have a new colleague at our department, Trifan, who is actually able to directly inject cells with genetic constructs. So to use some micro-injections. And so I hope that we will collaborate in the future. And instead of using adenovirus technique for transfection, which is not available here, we would rather use injection as the means to actually change the expression profile of the proteins. So you can see here 3 conditions, 3 different YAP construct that were expressed in the utricles. One was wild type. And here you can see on the bottom BRD USA that shows you whether supporting cells were able to re-enter cell cycle. And you can see that normal wild type construct, its expression didn't actually confer any regenerative capacity to the supporting cell. But this specific construct, again, so it was specifically designed with purpose, actually did. So, and I would like to illustrate the point with this slide is that we were able to manipulate pathway and to change the ability of supporting cells to re-enter cell cycle, which is really important step in our process of understanding how we can actually make supporting cells to regenerate. So if you have any questions regarding that, you can ask me later. And I just would like to say that a lot of details about these experiments and more specifics can be found in this paper. If you're interested, I refer you to. And so last but not the least, I would like to say that I started similar experiments here at Nazarbayev University. So I'm using explant cultures. And so what's actually a new technique I'm trying to establish here, it's called proximity ligation assay. So one problem that most researchers have when they study those sensory epithelia is the very small size of the tissue. So you're not able to do any biochemistry with it. So it's just size is so small that you can forget of using any biochemistry technique, and which normally people use to study protein-protein interactions. So for this reason, to study protein-protein interactions that are important for this cell cycle reentry, we are planning to use proximity ligation assay. And the principle of this assay is shown here in this slide. So let's imagine you want to study whether two proteins interact with each other. For example, one of those proteins can be YAP, transcriptional coactivate, another one can be, for example, TAD, which is transcriptional factor. So if they really interact, they will be in close proximity to each other. So when you label those two proteins with antibodies, which would specifically recognize, and those antibody probe can be linked to some specific oligos, then if they're in close proximity, then if your samples with ligase, your ligase would be able to ligate these oligos together and create the circular DNA. So, and once you created circular DNA, you can use DNA polymerase and run a DNA polymerase reaction by using a substrate, some fluorescently labeled oligonucleotides. And as a result, you will get at the site specifically where these two proteins were colocalized and interacted with each other, would be able to detect it inside of the cell on the level of one single cell. And that's quite a remarkable technique because it allows you to assess the two proteins interact with each other on a single cell level. And that's what I'm trying to establish here. So here you can see explant cultures which were plated on this rigid surface. Those are actually sensory epithelia. And you can see they started already to spread, and the cells started to stretch. And we know that at this leading age, at P0, this is actually the site where cells already ran the cell cycle and started to duplicate the DNA. So nuclei can be labeled with specific Hoch staining in these cells, Hoch stained heterochromatin, and I hope you can appreciate that you can easily stain nuclei of those cells. And in this magenta signal, we actually, that's the signal from proximity ligation assay. That's assay which we use to study TAD-YAP interaction in my lab. And I hope you appreciate that on the single cell level, you can see that in some of the cells, there is interaction, in the nearby nuclear interaction is absent. So here specifically, that's, for example, hair cells, there is no interaction because there is no YAP. But here are some potent cells, and you can see there is no interaction. And we also know that in these cells, they have not entered cell cycle yet. So I just wanted to demonstrate to you guys that those techniques, even though they're extremely complicated, we are quite capable of doing them here at this university. And so I'm pretty confident that they will allow us to study a lot of molecular processes on this kind of single cell level. If you have any questions, I will be happy to answer them. I think I should stop. We don't have much time left.
Okay, actually, if you have like 5 or 6 slides, we can still go over them, or we can stop here.
Well, we can stop here. I can just mention that in the future, I would like to focus more on molecular mechanisms that restrict proliferation capacity. And so we will use this model to study which molecular pathways are involved, which protein-protein interactions are involved, whether some of kind of modifications that we were able to detect in the past play a certain role or not. But yes, so major focus would be whether we would be able to alter those molecular mechanisms to actually force those supporting cells to enter the cell cycle and to help to regenerate tissues.
All right, so then we're going to go straight to the QA session. If someone has questions, you can switch on your mic, or you can write your questions to the chat. So we're going to wait for a few minutes. And while people are thinking, while they're processing this informative presentation, I'm going to ask you questions myself. They're going to be like general questions. Do you have some preferred skills or knowledge that you would advise students to acquire before they actually write email to a professor or ask someone to take their in their lab?
Not really. So when you join the lab as undergrad, and here we are talking about undergrads, nobody expect that undergrad joins the lab being proficient in certain technique. We don't expect it. At least I don't expect it. I initially assumed that when student joins the lab, I will spend 2-3 months teaching the student, and there would be a waste of reagents. And the question is how much reagent your student will waste to the trash before he or she would be able to learn technique. What I would like actually to mention though is that I would prefer students who take not easy courses. So for example, a lot of biology students are shy and they don't want to take Calc 2, Physics 2, or maybe Organic Chemistry 2. So I, as a professor, would prefer that you will do these courses rather than you learn certain techniques. And these courses, they really help to make certain brain connections, and students who took those courses, they do far less mistakes when they do them, and they learn much faster, and they normally succeed better. So for example, out of 4 students who actually 4th-year students right now who were in the lab, I work right now with Tamiris. And I can say Tamiris, she's from chemistry department, so she took all these courses. And the other 3 students that I had, they were from biology department. So they didn't took those courses. And I should say she's far more advanced than they are. So she's doing far much better job in the lab. I think it's just this scientific background really helped her. That's my opinion. Maybe it wasn't those courses, but something else, but that's better for some reason.
All right, and following up the question about students in your lab, how would you advise not to ask a professor for joining his lab? What not to do?
What not to do? Well, so it's hard. So I don't know, but so it depends on the professor. But what professor really doesn't want is the sloppy student who actually has no interest in the lab. So very often when student comes to the lab and says, I want to do research, they tell you a lot of nice words like I'm passionate, and then they disappear for 3 months. And so which leaves us with question, were they really passionate? So normally if you're serious about joining the lab and doing research and I would recommend students just initially assume you will need to work in the lab each day. So if you tell your professor that you do each day and you disappear like for some periods of time, then no. So, and that's probably the only thing. So the professor wants to see that for your research is on the list of the priorities, on the top of your priorities. If professor is that research is not your priority and you have others, and most professors would be really reluctant to take you into the lab.
Okay, thank you very much. And maybe let's do the last question before we finish. It's just like a quick answer. I suppose, what is the most common misconception about undergraduate students doing research, in your opinion?
Misconception by who? By professors or by students?
Among students about research.
Amongst students. So a lot of students at Nazarbayev University specifically think that research is something when they come to the lab and professor gives them the protocol and explains them steps 1, 2, 3, 4, here at 3 microliters, here at 10 microliters, and they follow the protocol, do it nicely, and that's research. It has nothing to do with research. It's more like, I don't know, it's like what pornography has lab. So it's really not research. You just followed protocol. There was nothing you gained from it. So research is when a student actually is very mindful about what he or she is doing. It requires read a lot of literature. So I would say students need to read at least 1 paper per week, maybe like if you're strained for time, be 1 paper per 2 weeks. But if student doesn't read papers related to the topic, a student will have no ideas and they would not be able to actually even gain what they were able to gain from this project. And unfortunately, students don't do this here. So I had several students who did, and I can name them. So Arai did and still does. So she's reading. So Janibek did a lot. Tamiris is reading. And so those students are really engaged in research. So if you just come to the lab and you just follow protocol, even if you do it nicely, in United States universities, you would not be included in the paper because it's not research. You didn't do any mental contribution to this. So yeah, so research is when you actually processes everything through your brain, you have your own opinion, and you are able to find mistakes and actually have your own hypothesis how to do this project. So if you don't have it, you didn't do research, you just kind of copy paste something. And that's, well, normally after that, most professors in the United States, they would not add your name to the list of courses.
Okay, I think everybody gets something new and something very important for their future attempts to be engaged in research. All right, again, thank you very much for joining us today. It was a very interesting session. I think we will ask you for some pictures and for links to the articles that you mentioned today, so we can share them with students. Also, I want to remind everyone who attends the sessions that there was a recording. It will be uploaded to YouTube where you can rewatch it, maybe then ask some questions. Okay, so we're going to stop here. I'm